Welcome to Maricated Knowledge Channel, your favorite resource point for medical and anatomical knowledge. Today, we will be looking at a very important topic: cell organelles. Organelles, while having similarities to organs, differ from organs. The similarities lie in the fact that organelles are parts of the cell performing specific functions just as organs are to the body. In cell biology, organelles are specialized subunits usually found within the cell that perform specific functions and are important to ensure the health of the cell. Before delving deeper into cell organelles, it should be noted that cells are broadly categorized into two: the prokaryotic cell and the eukaryotic cell. The prokaryotic cells are the virus, bacteria, etc., and these are cells that lack a membrane-bound nucleus and also lack intracellular membranous organelles. The eukaryotic cells, on the other hand, are found in plants, animals, humans, etc. These cells possess a membrane-bound nucleus and also possess membranous organelles. Another noteworthy point is the parts of the cell. Depending on the study material or guide, the cell is divided into 3 or 4 main parts. This is an eukaryotic cell, and while it might not be well detailed, it will suffice me to show the parts of the cell. The first part of the cell is the cell membrane. I am leaving the cell wall aside based on the fact that many eukaryotic cells lack a cell wall, and in such situations, the cell membrane is the first part of the cell to be encountered. After that, we have the nucleus, which is the second part of the cell. The nucleus is made up of different components such as the nucleolus, the nuclear pores, the nucleoplasm, etc. The third part of the cell, which is the last part in some study materials, is the cytoplasm. The cytoplasm is said to be the part of the cell which is internal of the cell membrane and external of the nucleus, including the organelles which are found in those locations. But some study materials say that the cytoplasm is only the fluid portion which is found between the cell membrane and the nucleus. In such a case, this will mean that all these organelles seen here make the fourth part of the cell, the organelles. Back to cell organelles. Cell organelles, based on their location, are broadly categorized into two: the intracellular organelles and the extracellular organelles. For this lesson, we will focus on the intracellular organelles. The intracellular organelles are found within the cell and are grouped into two: the membranous organelles and the non-membranous organelles. For this episode, we will only look at the non-membranous organelles, with some examples seen on the screen.
Like, subscribe, and share if you have been following the lesson. Also turn on notifications to get informed about new episodes. You can also drop your comment in the comments section to ask questions or let us know where you think we made a mistake or could have elaborated more. Your inputs are very much appreciated.
Non-membranous organelles are those organelles that are not bound in a membrane or bilipid layer. They have contact with the cytosol due to this. The cytosol here refers to the fluid part of the cytoplasm. On the screen are diagrams of a prokaryotic cell and an eukaryotic cell. It can be observed that while the eukaryotic cell possesses both membranous and non-membranous organelles, the prokaryotic cell possesses only non-membranous organelles, and this is because prokaryotic cells lack membrane-bound organelles. The diagram seen on the screen has the following parts to be non-membranous organelles: the ribosomes, the centrosome, the nucleolus, the microfilaments, the microtubules, and the intermediate filaments. The nucleolus is a spherical body found within and the biggest structure in the nucleus. It is the site of ribosome biogenesis, and its primary function is to produce and assemble the cell ribosomes. It is also where transcription to RNA occurs. Transcription is the conversion of DNA to RNA, and translation is the conversion of RNA to proteins. The ribosome gets transported by the nucleolus to the cytoplasm after it has been assembled. It appears flattened and spherical in shape when viewed under an electron microscope and is composed of two subunits, a larger and a smaller subunit. The ribosome is made up of RNA and proteins and serves as the site for protein synthesis. The ribosome does this by reading the messenger RNA sequence and translating the genetic code into specified amino acid strings, which grow into long chains that fold to form proteins. The messenger RNA gets bound to the smaller subunit where it is decoded. It is then moved to the larger subunit where the amino acids get added. The two subunits are joined together by the interactions between the ribosomal RNAs in one subunit and the proteins in the other. The ribosomes in the cytoplasm are either found free-floating or attached to the endoplasmic reticulum to form the rough endoplasmic reticulum. The centrosome is just two centrioles placed at 45 degrees to each other. It is a cellular structure that is located near the nucleus of the cell in the cytoplasm and is involved in the process of cell division. Its function in cell division is to pull the chromatids apart and organize the microtubules to provide structure for the cell. The spindle fibers also develop from it, and it also helps determine the location of the nucleus and other organelles within the cell after cell division. The centrioles are made up of 9 triplets of tubulin protein or microtubules arranged in a cylindrical form. The centrosome is found only in animal and human cells. The microfilaments are double-stranded molecules of polymerized fibrous actin, present in bundles and form a 3D intracellular network. Actin is a protein and it is the most abundant protein found in cells. The microfilaments are made from globular actin that are assembled into 2 long strands that twist together. They play major roles in cytokinesis, cell membrane motility, endo- and exocytosis, secretion, and vesicle transfer, and in structure and support of the cell. Endocytosis is the process of taking in material by enclosing it in a vesicle. Exocytosis is the opposite process, where the vesicle fuses with the plasma membrane and expels its contents to the outside of the cell. They can be found in both prokaryotic and eukaryotic cells and are very important to a cell. The microtubules are hollow, straw-shaped filaments that are made of linear protofilaments of alpha and beta tubulin. Tubulin is a protein. A protofilament is an ordered row of tubulin dimers. The microtubules function in mitosis where they play major roles in the formation of mitotic spindles. They also function in cell motility, maintenance of cell shape and intracellular transport. They are found in all and only the eukaryotic cells. They give structure to cilia and flagella, but only those of eukaryotic cells. The intermediate filaments are made of dimers that come together to form tetramers, and the tetramers assemble end-to-end, overlapping to form long strands, thereby making them appear as solid, rope-like fibers. They are made of different proteins. The intermediate filaments work together with the microtubules to give support to the fragile tubulin. Intermediate filaments help anchor cells together, providing strength against tension, and also serve as shock absorbers for cells. Intermediate filaments help anchor cells together, providing strength against tension. Intermediate filaments are majorly found in animal cells, and although the proteins have been found in higher plant cells, their functions are yet to be understood. The microfilaments, the microtubules, and intermediate filaments are the 3 cytoskeleton of the cell and are all non-membranous. This draws the curtain on the non-membranous cell organelles, and we will learn about the membranous organelles in another episode from this channel. So, if you haven't turned on the notifications for my channel, do so now to avoid missing out.